Hello, and welcome to this overview for lab number 6, cardiovascular effects of exercise for Biology 206 lab, human physiology lab. As always, kind of a set of objectives to kind of help focus on what we feel are important in the activities that we're doing this week. So in this week, we're going to be looking at cardiovascular system functions. And so we'll start out looking at an electrocardiogram, an ECG, previously known as an EKG. And so what I've got on here are just a couple diagrams showing different ways in which we can take measurements using the electrocardiogram. Now, a very simple way is to use 4 leads located on the limbs. So, leads on the left arm, the right leg, and the left leg. And basically what we have with these electrodes is the ability to measure the intensity, the magnitude of electrical changes within the body associated with the heart properties, the heart conduction, the heart contraction. And so we can see kind of both the magnitude of the changes with the heart contraction as well as the direction that it's moving. And so whether or not the electrical charge is moving towards the lead or away from the lead. And so the 4-lead system is a nice simple way to look at an ECG. If you're in a clinical setting using an ECG as a diagnostic tool, you're often going to use a 12-lead ECG. And what you can see here is that you still have a lot of positions associated with the arms, with the limbs, but you're also going to have a series of leads that are going to be located in the thoracic area to more accurately kind of measure what's going on in terms of the electrical activity associated with the heart. Now, in general, even though we're using a 12-lead ECG in a clinical setting, they're going to be basically comparing 3 leads at a time. So measuring kind of changes in kind of the voltage differential and the direction of those voltage changes in relationship to the body, and then using these different leads. Now, in our laboratory setting, we're actually going to be using kind of a modified 4-lead. We're actually going to be using a 3-lead system. And so kind of similar to what we did with the EMG lab, the electromyograph lab a few weeks ago, we're going to use an interface attached to the power lab system. We're going to have a series of leads that are coming off. And so we're going to have the earth strap, the grounding strap, which is going to be located on the right ankle. And then we're going to have the positive lead attached to the wrist on the left arm. And the negative lead located in the wrist area on the right arm. And this is going to give us a fairly reliable and very easy to read ECG. Now, like we've seen in the past, if these electrodes are a little bit loose, or if you haven't kind of cleaned the skin and used some electrode gel, you may not get a good connection. And so you want to make sure that the leads are attached, that you've got a good quantity of the electrode gel that doesn't interfere with the ability of the electrode to stick to the skin of your subject, but still allows for the picking up and the reading of these electrical signals. Now, when we look at an ECG, basically we're looking at a trace here that is going to be appearing on your lab chart view. And within this are going to be a series of identifiable waves. So we've got a P wave here kind of at the start of the contraction. This is going to represent the atrial depolarization. And then we're going to have a little bit of a gap here between the depolarization, the contraction of the atria. And then once the atria have been contracted completely, then we're going to have the ventricles depolarize, the contraction of the ventricles within the QRS wave. And then what we've got over here within the T wave is kind of a repolarization of the ventricle. And so you're going to see kind of a going into a relaxation phase and the heart kind of resetting in order to have another contraction. Now, when we take a look at this, again, there are a lot of measurements that we can take a look at. But mainly the ones we're going to be focusing in on are going to be associated with the intervals that kind of tell us what's going on within the region of the heart. In addition to that, we're going to be looking a little bit of cardiac output. And the cardiac output, basically how much blood is going to be pumped out of the heart and distributed to the body within a minute of time. And so if we take a look at that, a variety of factors, and again, we're going to be covering this in lecture as well, but we're going to have kind of the number of times the heart is beating within a minute. I'm sorry about that. And so increasing the heart rate would be pumping the heart more and having more contractions and more kind of potentially more blood being distributed throughout the body. And then we have a stroke volume associated with it. And so kind of the amount of blood that's going to be pumped out with each contraction of the heart. And so if we take a look at this cardiac output, the amount of blood that is going to be distributed throughout the body within a minute of these heart contractions are going to be a factor of how many times the heart is beating, so the heart rate, times how much blood is going to be kind of ejected from the heart or kind of expelled from the heart with each contraction. And so within lecture, we're going to be talking about some of these different values, but we're going to be looking at this in a limited extent within the laboratory this week. Now, keep in mind that a big role of the arteries within our body are going to be to kind of regulate the blood flow. And the way they're going to do this is by, with kind of varied innervation, autonomic nervous system innervation. We're going to see that under some circumstances, the arteries will be constricted. And so basically activation of the smooth muscle cells within the wall of the artery can result in a narrowing of the blood vessel that's going to increase the resistance and make it more likely that the blood is going to be shunted off into another region of the body. And so, we've talked about this within lecture, the difference in the blood flow associated with kind of stimulation under the sympathetic autonomic nervous system, the fight or flight response is going to be very, very different than the blood flow distribution during the parasympathetic rest or digest and control. And we'll look a little bit about that as well. Now, if we look specifically at what we're doing in lab this week, what we're going to be working with, again, going onto Canvas. I should probably go over to Canvas so you can see it a little bit more accurately. And so pull this up. And so this is what we've got for this week's module, the Lab 6 module. We'll have a Zoom meeting link for the remote students. This video will be posted here. And then we've got the ADI instructions, some more detailed instructions on how to set up both the Power Lab system as well as using the lab chart software for analyzing your results. We've got a PDF version of our lab activity. Again, because you're going to download a template off of Google Docs, save it onto your computer if you're going to use Microsoft Word, or save it into your Google Docs folder if you're going to use the Google word processing system. So there's a PDF Word version in case this version gets edited down and you lose parts of it. In addition to that, I'm including a diagram showing the ECG parameters. So if you click on that, it's going to give you to, or take you to what I think is a very nice diagram, identifying where these different waveforms are being, where the different waveforms are on an ECG, as well as where we start and where we end on some of these important intervals and segments that we're going to be taking measurements from. Again, once you've done with the lab, you've completed the questions down there at the end, you're going to be able to submit your lab report here. Again, hopefully using the correct naming notations that we had talked about before, as well as if you've got some good data, if you're willing to share that with me. Again, this would be helpful for us to have backup data sets in case there are problems with the lab in any given week. As always, some additional resources from when we had the the lab online. So a nice YouTube video talking about the cardiovascular system responses to exercise. If you're going to be analyzing the results outside of the lab, you want to make sure you take the ADI-CHT files with you or email them to you. But if we have difficulty with the equipment, we have a mock data set for both the first activity and the second activity that we're going to be doing. And so ECG in general in relationship to exercise, and then the hand exercise module as well. And so you basically, you'll download your template, work on the lab. And so the first activity is going to be using the lab chart system with a 3-lead ECG. We're going to start out measuring an ECG and blood pulse rate at rest. And so we use the ECG electrodes as well as the pulse transducer. And there'll be a chart in which you'll go through and take these measurements. And again, refer to that diagram because that'll help you kind of determine where it is that you need to be kind of measuring as you're going through and taking this data. Again, if there are problems with the data, your lab instructor may tell you to use the mock data that's available on Canvas. So once you've been able to get good readings at rest, you're going to have your subject exercise like we've done before, and then come back for the second activity. And in that second activity, we're basically going to do the same thing we did in the first time. And so, don't pull off the electrodes, maybe unplug them, let them hold the lead as they're exercising. And then when they return to the lab, just plug those leads back in to the Power Lab system, and then start taking measurements at about 0. So as soon as they get back, about 30 seconds and about 60 seconds of recovery. Again, looking at the same basic measurements. Again, we've got some mock data that's available if there are problems using the equipment this week. The third activity is the ECG and blood volume pulse after hand exercise. And we're actually going to be doing it before and after hand exercise. And we're not going to be focusing in on the ECG with this activity. So you're going to be able to take the ECG electrodes off, but maintain using the pulse transducer. What you're going to do is start out, again, using the system, taking some measurements at rest. And then when you've got some good measurements, then stop the system, allow the subject to exercise their hand, basically squeezing their hand for a period of time. And then when they're done exercising, come back, start the recording, and then take measurements of heart rate and mean pulse amplitude at 10 seconds, 30, 60, and 120 seconds of recovery. Again, if there are problems with the system, we have mock data available. Once you've completed the lab activities, as well as the questions at the end, we're asking you, again, to use the naming convention that we've talked about before, last name, first name, lab report number 6 in this case, in case we have to download them to our computers to kind of comment on them and grade them. So please submit this at the point on the Canvas site that we've identified it. And if you're willing and you've got good data, if you could kind of upload some of the ADI-CHT format files here, this just gives us a greater library to work with as we're going forward within the course. As always, your lab instructor may have additional information available to you. And so please see if they have any changes or updates for the lab for this week. As always, if you have any questions, please do not hesitate to reach out to me. Thank you and have a great week in lab day.